Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted your audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speakers your questions from the chat window. Your questions in the chat window will only be visible to myself and our speakers. So thank you for joining us today. It is now my pleasure to introduce our speakers, Burkhard Joksch and Stuart Tindal from Sartorius Stedim Biotech.
Okay, thank you very much. So our topic today is actually multi-product facility realized by modular FlexAct platforms. So we want to discuss single-use manufacturing and how it's evolved and what we've learned in the last 10, 15 years as it's been more and more adopted within the biopharmaceutical industry. I think one of the first parts is that we can't really adopt a copy-paste attitude from stainless steel to a single-use facility. There's a lot more available, flexible manufacturing options, degrees of freedom that we can adopt within this new manufacturing technique. When we think about how the modularization we can have within the consumable sets, the wetware and components like that, we can be more flexible in that degree. And when we have closed systems, we can then move into more open ballroom or dance floor concepts where we can have technologies, hardware and wetware moving around the facility in order to facilitate different scales of manufacturing and different product process flows. We can obviously see how this adaptive technology can cover the different capacity needs of different products. We can also see how it can adapt different scales and volumes of the materials needed. And then we can also look at the different schedules or process flow order where different technologies can come in. And therefore, that's really sort of giving the toolbox of technologies really to be able to do multi-products. And when we add in our understanding about how we are developing technologies in a modular platform, and these become standard, and these instruments can plug and play into new technology facilities. So I want to look at the case of the FlexAct platform. And this is a modular platform for both upstream and downstream processing unit operations. We can cover media preparation and cell removal clarification from an upstream perspective, and then moving down through the different unit operations and the buffer preparation needed for DSP. We can cover the cross-flow filtration, virus inactivation, and virus removal. And these are the standard unit operation packages this technology can actually offer. They're built up with different sequence and recipe components that we can string together in a configurable sense and rebuild them in different orders in order to take different unit operations later. Of course, these technologies, this FlexAct platform can connect into customers' facilities, into the distributed control systems, manufacturing execution systems, and its data that it's generating going into multivariate data analysis. So these are the powerful components that with the hardware, wetware, and the software modules, we can bring together different unit operations standard, as well as in the future, different building blocks for different components. So my next part is what I want to understand is, are you actually planning something in the future to obviously enhance or expand or change your bioprocessing pilot, clinical or commercial production capacity? You can answer yes or no. Okay, thank you very much for your responses. So first, in regards to planning a facility, you really want to have a conceptual design. Again, going back to obviously stainless steel, copy-paste from there, and you have your zoned rooms where you're piping liquid from a buffer prep into your downstream facility, or your media prep piping it into your upstream bioreactors. In that sort of sense, you're moving liquids over long tubing, flexible tubing lines. in new facility designs or more open, where we're moving material in its totes or its pallet tanks and bringing the liquids to the actual unit operation and utilizing it there. Obviously, equipment can move back and around. So as we move to a more dance floor, ballroom concept design, we have more technologies in more rooms, or less rooms, apologies, or bigger rooms. So this obviously enables a lot of flexibility in how you're moving the technologies around and how you're connecting them, and obviously be able to do different things. But we'll obviously look at the segregation beyond the viral removal in post-viral, and that pretty much remains the same within in a multi-product single-use facility. But we really want to understand a little bit more about that capacity requirement. So the next part here is how much product would you be considering actually producing within your single-use downstream processing batch? Okay, so capacity there is obviously an important thing. So if you're really sort of looking at very high capacity, so 15 kilogram batches of product of monoclonal antibody, in one particular case, you probably have a high system utilization. On the contrary end as well, that maybe you have a low productivity, you're only maybe half kilo or 1 kilo batches, then maybe you're trying to facilitate more utilization in systems. So the part here is with the FlexAct Modular Platform is really being able to enhance that utilization of system. So we can say that we can have two FlexActs, just as a particular connected case with virus inactivation and virus removal, and they're sequentially within the process flow. We're processing via the two-tank virus inactivation and directly into the virus removal. Perhaps you're scheduling this directly one after the other within the process, or you might have a different shift performing one unit operation and the second shift performing the next unit operation. And if you've got the high utilization, then great, you can use that. But if you're moving it down into having lower productivities and you've got lower processing requirements, then maybe we can actually start to share systems in sequence. So one FlexAct can do both jobs because software-wise and the modular design of the technology, we can equip the same FlexAct in one way, take down all the equipment once we've finished that unit operation, and re-equip it to do the next unit operation when it's loaded with the software. Equally, in that particular part, with this higher system utilization, we can also minimize the downtime, because if in the unlikely event that one of the systems malfunctions, each of the other systems is able to be its own backup partner. So moving on, I want to look a little bit in regards to a case study of one of the unit operations, cross-flow filtration or tangential flow. Looking at the individual unit operation, we can cover a huge number of variants within the unit operation. Just some of the variant management that we can cover is within the filter options, and this is membrane material, molecular weight cutoff, or we can even add some third-party filters within the design space. Process control strategies, whether we have temperature control, pH, conductivity, in-bag or in-line, and we can look at the permeate tubing diameter coming off with the flow pipes and the pressure pipes and the retentate permeate flow constriction. There's a huge design space that can be configured from the wetware to the hardware, covering our recirculation volumes of down to 4 liters and right up to 200 liters, and feeding in product from 5 to 1,000 liters. But there's a huge design space that is built up over the number of years that we've been designing cross-flow systems, as well as moving into single-use cross-flow systems. But that's just that's the two components on the hardware and the wetware. But with that massive degree of automation, we really have to have a very good ability to handle that automation. And this is where I'm going to hand over to Burkhard, and he's going to explain how the automation handles this variability.
Yes, thanks, Stuart, for giving that over. Actually, to recap, we have 6 different applications, but even in one application, we see 30K variations. That is a lot of different variants which we have to scope in there for the automation. And I take the picture back where Stuart was introducing stainless steel to single use. In stainless steel, we had one dedicated plant, we have one automation set up, and we did probably a little bit maintenance. In this case, single use is with this flexible setup is calling for a different kind of automation. So we need to have somehow an automation flexibility in this case, and so that is based on the different process configuration which you have seen before. Of course, you have to change the different applications, the buffer preparation, cross flow, virus inactivation, virus removal, etc. As you have done so, and also say what kind of volume you are processing and what kind of sensors you are activating, using, then the automation is activating the right sensors activated, and also the controls attached to that, they are activated or deactivated. And at the same time, we pre-parameterize the sequence and recipes. For example, the filtration is done for big volume, and we are changing the PID control of the filtration step. Also, we will provide recipes, unit recipes for the specific unit operation, which is fully set up, so you can really run that immediately after that, and also have the HMI for your specific setup being changed and adapted to that. So we actually put down here the automation pyramid, and already scoped out what is the limitation of that, of our local automation. And we have to integrate somehow into the upper world, of course. And we would like to know what kind of integration do you have in your case, and is it Siemens, is it Amazon, or Rockwell? I'll leave it for you for a moment. Okay, thank you. So we have quite a good process through the different system. We have Siemens, we have Amazon and Rockwell, and also others. This actually says what we actually feel. Also, we are focusing on our smart modular package unit, of course, of the local automation, scoped by the yellow surrounding. But we have to integrate well into different SCADA and DCS systems, distributed control system, which are out there, which is probably Siemens, Amazon, Rockwell, and also some others. And we do that with a range of interfaces. We have OPC UA, we have classic OPC, so we can integrate this well into PI system, process information system. But we also have lower level interfaces like Profinet or Modbus TCP/IP for doing integration on a low level. And the scope of the smart modular package unit is actually to integrate all the single-use sensors, the actuators, and the pumps, the valves, etc., form also the basic controls and provide that in sequences and recipes, so filtration steps, mixing steps, but also a full recipe for the buffer preparation or for the cross-flow operation. And integration-wide, we do different levels of integration, and shown here and are the 3 different entry points. One is actually for remote I/O, where you simply use the actuators or sensors and write a value or read a value from the sensors and do all the rest in the upper level. We have also the entry point for the sequence level, where you simply use a sequence, in this case, for example, the pH inactivation, which is already programmed in our system. You parameterize that for the high and low pH and set, for example, the inactivation time and then execute it. Or you choose to actually, from the top level, execute a recipe. You select the recipe which is inside the system. You can have different recipes in there, for example, buffer preparation for different buffers. So you select the volume which you want to produce, and also some other parameters. In this case, we choose pH and execute the recipes from the top. This has different benefits, and also there are different scenarios in the world. Sometimes a lot of Delta V customers and also Siemens customers are doing the endpoint 3, the remote I/O. But on the other hand, you have to do a lot of integration work on the upper part. Okay, I have to go on. So what do you think, what kind of entry or what kind of integration scenario would you choose in your facility? Is it more recipe level or is it just remote I/O? I'll leave it for a moment. Okay, thank you. It's quite nice result, because a lot of you choose recipe level, which is, to my knowledge, out in the world, is a lot of customers who do remote I/O integration, and I'm happy to hear that, because there's a lot of time to do for the integration on a lower level. So we compared here the remote I/O tasks which you have to do. You have to do some function specification of the system if you do that, and software design specification, the model design configuration and specification for the different control modules, equipment modules and phases, etc. If you do that all in remote I/O, and you see some topics for the modular integration are actually reduced, and our statement, and we have done that together with some cooperation partner, is that we'll reduce the effort for integration by 50 to 75%, and that's a huge effort reduction, which will decrease the building time of your facility. So we already come to the conclusion, we would show to you today, and Stuart started with that, that we have a modular design of the FlexAct platform with all the different hardware, the consumables, the wetware components, and also have a configurable software. And by this, we enable the adaptation to a process change and also product change quite easily. And because we have the coded, pre-tested and pre-qualified sequences or even recipes included in our system, we will have a lower integration effort. And by this, we will reduce capex reduction, we will have a faster product changeover, and also the time to build a facility or come to a process qualification will be reduced. So by this conclusion, we just open the question and answer session, and we're happy to hear.
Yeah, we thank you for your attention, and if you want to populate a couple of questions, if you have any, into the question box, but I think we hand back to Leah as well.
Yes, thanks, Stuart and Burkhard. So the first question is, what scales of bioprocessing would these modular systems be able to manage?
So our current, let's say, design space that we're covering, we're really looking to manage the 2,000 liter production bioreactor. So basically facilitating the media for the production bioreactor, from media preparation and then cell clarification. So with the different range of depth filtration or body feed filtration, we can actually manage that 2,000 litre within a certain limit of cell density. And then throughout the production capacity of that bioreactor, whether it be 2 gram, 3 gram per litre, we can manage that process stream right down to the final cross-flow filtration preparation into the filling either of drug substance or filling over to the fill machine. Okay?
Okay, so on one of the slides you spoke about interchangeable or flexible equipment. Is this a plug-and-play concept that works?
Yeah, thanks for the question. It's a good question. We are working together with the BPOC group on the plug-and-play concept, and also in the Namur Z4E approach, the MTP, the modular type package. Yes, it is somehow an approach to make it plug-and-play, or to make it at least interchangeable between between different systems. And also with the standardization envisioned in the MTP and also in the BPOC plug-and-play, it is an interchangeable system between different vendors. That is the goal of this concept and this phase integration.
Okay. So one last question before we get going. If we didn't get to your question, the questions will be passed to our speaker and they can follow up with you directly. So the final question is, how do you see the integration at the recipe level with Delta V working?
That's a good point. I think with Delta V we will see recipe integration is a little bit more difficult, and we are in constant talk with Delta V engineers to do that. In the meantime, I think at the moment, a lot of people will do in Delta V the remote I/O integration, but you see already Emerson Delta V opening up with some concept already to make an interchangeable system happening. So you put the intelligence down into the system. They have already something, and we are also in talk with them to increase the capability to have recipes in the system below.
Okay, thank you, Stuart and Burkhard.
Thank you.
And thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you at future Bioprocess International Ask the Expert webcasts. Look for the announcements in your inbox. Goodbye.
Goodbye.
Goodbye.